So our second speaker is myself, and I think Dr. Welch gave a great introductory, on what metastasis is. And like I mentioned, I'm an assistant professor at USC, and my background is in neuroscience, and my research is in brain metastasis. But today, my goal is not to talk to you about brain metastasis, but basically bring you into the research lab, because I know that so many of you are curious about what we do, all of us on the panel, and have never seen a research lab or some of the basic concepts of how we do experiments. So that is going to be my goal for today. So Danny brought up, gave a great lesson on the biology of cancer and metastasis. I'm just going to, not going to be redundant, but just to say, the word cancer, I mean, you got to go back to almost, 400, BC, where Hippocrates noticed that these cells that were not normal had the shape of basically crabs. And that's actually what the word cancer means, is because these cells are shaped like little crabs, and they have, these long feet and hands. Now, with respect to breast cancer, the terminology, we always hear HER2 positive, ER, PR, triple negative. What does that really mean? So the way tumor cells get to function and they work is they communicate with little, antennas on top of the roof of their cell. So the house, if you think of a cell as a house, then you have antennas, and these antennas could include what are called receptors, and one of those receptors is the HER2 receptor, which is a subtype of breast cancer. With respect to the other subtypes, so ER and PR, these are estrogen and progesterone receptors. These are necessarily not on top of the cells, and I know what I just mentioned, but they're more of antennas within the cell. So think about it as sort of like the Wi-Fi that we have, inside our own homes. And the elusive triple negative, all that means is that cell does not have either the receptor on its roof, the HER2, or the internal Wi-Fi signals, which is the ER and the PR. And that unfortunately makes it very elusive, because we don't have what's called a targeted therapy for it. We don't know how to specifically go after that tumor cell. Danny mentioned this very well. Cancer is more complex. This is, an image of, what the old pathology books and anatomy books showed of what cancer is, that it's just basically two normal cells that start dividing very fast. And we know that that's not the case, that they are not all the same as it is in this picture. And that the word there that I want you to remember is the word heterogeneity. And Danny mentioned this. It's basically very different. So it's like a melting pot of cells, which all come together. So why does cancer kill, or why does treatment fail? And this is the concept, one of the concepts of, tumor dormancy or hibernation. And Sirius Kajar is going to go through this concept later on, and Peter Kuhn and Alana are going to be speaking after me, and Dolores or Davizia are going to be coming. One of the main concepts is, it goes back to that word heterogeneity. So imagine a tumor cell is different color marbles. You have some purple marbles, you have some red, and you have some yellow marbles. Well, chemotherapy, the primary types of, or the current treatments that we do have right now, they'll kill the majority of the cells. So they'll kill all the purple cells. But what ends up happening is that these red and yellow marbles are basically resistant. They have the various characteristics of becoming resistant, whether they go to sleep, like hibernation, whether they're just very aggressive and they can beat out the chemotherapy. But these are the seeds of what the reoccurrence, or as well the metastasis, a metastatic tumor pop-up. These are the seeds for these tumor cells. And to go back to that whole world of heterogeneity, what Danny alluded to is that it's not a simple thing. Danny alluded to the freeway system in LA, or, think about it in New York, the subway system, or even Shanghai, the subway system there. And so what you see now is that it's not just the tumor cells that are doing all of the work. They are actually hijacking, our normal body's own, in this case, immune cells or other supportive cells, to help them grow. And so this is just one example from, our research when we do brain metastasis, showing that, tumor cells are sitting here nicely with brain cells, the neurons here. And they're actually, you see what Hippocrates called, the cancer cell, the crab-like. They are basically these little, hand and feet of the cells that are basically holding hands, they're grasping each other. And through these holding of hands, they're passing back and forth molecules that are either good for, the tumor cell to grow, or they are setting that environment that Danny mentioned. So metastasis, okay? Metastasis is, like Danny mentioned, is just a spread, but what does that physically look like? And this is something that is, I want to make it clear, and Danny did a great job of introducing this. But what ends up happening is that a woman will never, or a man, I should say, will never die of the disease within the breast. It's when those cells pick up and they go through our blood vessels and they start floating around and swimming through the pipeline of the body, and they land up in other organ systems and grow. So for example, when we say breast cancer cells have metastasized to the liver, it's not that the liver itself is cancerous now, it's actually breast tissue growing within the liver. The same concept goes with brain metastasis. So when my neurosurgical colleagues, they take out a brain metastasis from breast cancer, and I know it's somewhat of lunchtime wherever you are, but sorry for the analogy, but the brain is, imagine it has a jello texture and the breast is very fatty. And so it's really when they, in a cottage cheese texture, so when the surgeon goes into the brain, it's actually jello surrounded, that is, I'm sorry, it's cottage cheese that is surrounded by jello, because it's breast tissue growing within another area. Another concept is the circulating tumor cell, or liquid biopsy, what Peter Kuhn is going to be talking about, is that these tumor cells are now floating around our body. And also, not only are they floating around on our body, tumor cells, whether it's in the primary site, which is the breast, or in other organs, can be sending out these cargoes. And that's the extracellular vesicles with Dr. Divizio is going to be talking about. And these cargoes help promote, like Danny mentioned, the summer homes, of these tumor cells as they're traveling around. And so I took this from the headlines in January, from TMZ. It came out that in January. Then this is the misnomer that that we need to basically educate the community about, that breast cancer, when it travels or metastasizes into other regions, it's still breast cancer, but it's growing in different regions. So Taylor Swift's mom, that was the headline. Mom also battling brain tumor on top of her breast cancer treatment. Well, this is certainly not the case. It's not that it's that breast cancer, that she has a brain tumor on top of it. It's actually breast cancer that has traveled in other regions. And this also holds true for other cancers, and other types. I mean, we lost two great people this past year, Chadwick Boseman to metastatic colon cancer, and more recently, Ruth Bader Ginsburg to pancreatic cancer. And the term always is metastatic. So these tumor cells, whether it's breast or colon or pancreas, they leave their original site and they go to other places. So the key word is metastasis. Now, how do we look at metastasis in the lab? And I want to bring you into this. And so the way we, everybody imagines, cancer and the research we do, it's sort of like not like Breaking Bad, or it's not like, the things that you see on TV. So, and movies where, we get a DNA sample in the morning and then we unravel everything by at nighttime. This is, research lab. This is how a research lab looks like normally. And so we have, as you'll see, and a lot of my colleagues can attest to, we have our bench tops, we have our different, tools that we use, pipettes, we have all of our, grad students, postdocs, we have Post-its everywhere. And so I want to bring you into what does the research lab really look like, and how do we study cancer cells within a dish? So you've heard of this, studying cancer in a dish. How do we do this? So when our oncologist colleagues, they take out a tumor, whether it's, breast or lung, we grow them in these little Petri dishes and in the lab. How do we grow things outside of the body? Well, we have to mimic the body, and this is called in vitro or in cell culture. So the way when we get tumor specimens from the OR or biopsies, we work in what's called laminar flow hoods. like this. And so we have pictures here. This with inside of here, it's sterile because we want to, well, our immune system is protecting our body from bacteria and viruses. And we want to study the cancer cells, and we have to do it in a sterile environment so that the tumor, what we are actually studying is actually the tumors and not the fungus and the tumor and the viruses and bacteria that are floating within the air. So we do everything within this sterile environment and are in these what we call cell culture hoods. We grow our cancer cells in what we call media. So this is solution. So this is food for them. Okay, we give them nutrients. And if we're studying, for example, dormancy, we give them less nutrients. Or if we're studying, let's say brain metastasis, we give them nutrients that are similar to the brain environment, the food that our brain receives. And you might say, well, our food is food to our body. No, but what ends up happening is a different environment. So remember that word. Different environments have different nutrients. There's subtle changes that these cancer cells take advantage of. Finally, I said the word mimicking the body. We grow our cancer cells in something called incubators. So this basically it mimics the body's temperature. It mimics the body's carbon dioxide, oxygen levels. And so it's basically a vessel. So if you think about it, it's a vessel for these tumor cells. When we put our petri dishes in here, they grow in the right oxygenated and temperature, just like the body. Now, how do cancer cells look like? Well, these are 3 different pictures of 3 breast cancer cells that we have. And what's interesting is that they're all breast cancer, but they all look very different. Okay, and this is one of the other hurdles that we have to go through. When we hear breast cancer, we think, oh, all cells look the same. But they're not. They're from 3 different patients. They're all breast cancer, but they look different. And so imagine the genetics that's under there that is driving these cancer cells, how different they are if the cells themselves look by shape different. So how do cancer cells look like? So this is called the time-lapse video. So these are cancer cells just moving over about 2 days worth. We've increased its speed. What you end up seeing is that these tumor cells start what we call proliferate or divide. And you see these little tumor cells here that are floating around. Well, this is sort of them picking up from the dish and just floating away. And this sort of is what happens with metastasis. So you see this little guy that just came through, and I'll replay the video again. It'll just start floating around. This is, in a very simple way, what metastasis looks like, because these tumor cells are growing very flat and they're holding on to that collagen, for example, that Danny was talking about. And then they'll basically anchor off and they'll start floating around and they'll whiz off. And that's what metastasis is. And you see right here, my little prompter here. So how do these tumor cells, we were talking about the environment, and I'm going to use, one slide for brain metastasis to talk about this. Cells, remember what I told you, they talk back and forth through the antennas, okay? And these antennas basically capture molecules from their surrounding regions or within 2 tumor cells talking back and forth. So imagine if my kids, they were playing back and forth, they're throwing a tennis ball back and forth with each other. That's exactly how our cells communicate within our body, is that they throw little molecules back and forth, and those molecules basically tell each cell what they should be doing. So how do we do some experiments? So we talked about environments. And how do we do these experiments within the laboratory? So remember I showed you the dish here before? We can culture 2 types of cells. Now we have a little insert on top. We can basically culture breast cancer cells, and on the bottom, we can culture brain cells. And that's, so this insert up here is this right here. And we put this guy in this little dish right here. And these little balls here that were floating around are those signaling molecules that the breast cancer cells are basically going back and forth with the brain cells. And we can basically study what those are and how can we stop them from communicating with each other. We can also study metastasis. So remember, how do we study something that is floating through our body? We can study metastasis, what's called on a chip. And this chip is basically analogous to a computer chip. But what we do is we load up these little channels, what we call, with tumor cells. And these tumor cells now whiz through this chip. And based off of how that chip is, it basically can pick up different signals, different molecules. It can pick up different properties of those cells traveling through the channel. And we can test drugs this way. We could test resistance through this. So this is what's called a microfluidic chip, if you've ever heard about it. And it's very small, but that's the most amazing thing, is that you can load it up again with cancer cells and study different properties of it. So let's look under, some very, very simple assays of how do we look at things, in a cancer cell. So going from all the way from its DNA, how that DNA is basically made into RNA, and that RNA is in turn made into proteins. So what kind of techniques do we use? So if you think about, DNA and RNA in cancer biology, is that imagine, an author, you're sitting there, you're typing up a book. If we didn't have word and spell check and autocorrect, you would probably be making these little, spelling errors. These spelling errors are exactly what happens with our DNA or our genetic codes. As cells are dividing normally, sometimes there is error. These errors can misspell a word. And if they misspell a word, then the theme of the book or the page is now changed. And that's exactly what happens with cancer cells. I'm sorry, with normal cells. As they're writing their genetic codes, there are spelling errors, and that spelling error all of a sudden turns that normal cell into something it shouldn't be. It changes their storyline and that timeline. And so, how do we check the proofreading? We have machines that basically what's called sequencing, and it basically we can read through the DNA and the RNA and check to see what those spelling errors are. And based off of that, those spelling errors are genes that have changed that normal cell into a cancerous cell. And then we could study what those spelling errors are and try to figure out how has that changed the story of that book? And how can we stop that error from occurring or causing more problems within that cell? And so that word is called DNA and RNA sequencing technology. It's just proofreading. The next is, well, how do we measure these errors that happen in a cell? So imagine the gene HER2. We use techniques called qPCR, and we measure basically the RNA of HER2. And so in a normal cell, HER2 or that antenna should be very, it should be expressed at a very modest level. But in tumor cells, it's expressed at much higher levels. And when we quantify this, or we look at this through qPCR. Finally, we get the protein staining. How do we, so for that RNA, for that HER2 to actually work, it has to be changed into protein. It has to be physically changed into the antenna. What we were talking about, whenever we talk about DNA, RNA, it's located in the nucleus here. But that signal, that message has to be changed into protein, and it has to be now shown on top of the cell. We can measure what HER2 signaling looks like by, techniques called immunofluorescence. So it's basically putting an antibody, something that recognizes HER2, and attaching it to the top of that cell, and then that tagging mechanism or that antibody now shines and glows through a fluorescent technology. So for example, when you hear the word immunohistochemistry, or what pathologists look like all the time, is they look at tissue staining. Okay, and so tissue staining, for example, so this is HER2 amongst tumor cells that have been stained for right here. So pathologists, the way they recognize or they diagnose tumors is by staining procedures. So whenever you hear that word, think about this technology, immunohistochemistry. And this is a fluorescence way of looking at it, and this is what we call a non-fluorescence way of looking at it. We can do the same thing to cells individually, so they don't have to be in tissue. So these are basically images that I've taken, and you're looking at cells now. And the green here is the skeleton of the cell, and the red is sort of like is like the muscle-like, or again, the skeleton. The blue in every picture is the nucleus, where the DNA is occurring. And so now you can start characterizing the different types of cells. So this is, part of a brain cell. This is a breast cancer cell. And this is another breast cancer cell. And these are basically stem cells looking. And so so you can start looking at the landscape of cells in different ways. Finally, we can make 3D images of those. And so these are brain cells that we have. So these are neurons. We can basically put those pictures together and you get a 3D formation of cells coming. And this is done through what's called a confocal microscope. We can also separate cells. So we can take cells, for example, in breast cancer. So if you take a tumor mass, you can take cells that are HER2 positive and cells that are HER2 minus. So remember those marbles that I showed you earlier on. If half of those marbles have that HER2 protein on top of them, we can take through what's called the flow cytometer, and we can put these cells through this machine. And based off of if they express that protein or not, they get sorted into one pot versus another. And so that way we can just specifically study this, what we call population versus this. And this technology is called fluorescence cytometry. So I hope that you got, a little bit glimpse of what we do in the lab. I mean, this is, again, all of these techniques. There are many, many more techniques, but I wanted to teach you or show you just a little glimpse of what we do, in the lab. And what we call, what the cells like to call us is we are their paparazzi, okay? Because we like to glimpse into them. And so with that, I will end my presentation and stop the share. And I hope you've, got a better idea on this. And I will let our next speaker, Alana, go ahead and share, and I will introduce Alana. And she's sharing her screen.